these two, the two different bands will appear, and due to arrangement of the dark and light bands synchronously, the complete muscle fiber or the complete muscle bundle will show dark and light bands. Whereas in smooth muscle, the distribution of myosin and actin are irregular. That's why the dark band will be scattered. We cannot see striation. Yesterday I explained the structure of myofibril. Two jet lines, one M line. M line is present at the middle of thick filaments. These filaments are very thick. And jet line is present in the middle of thin filaments. The thin filaments are present like this. So wherever the myosin filaments are present, that appears very dark, that is dark band. Wherever the actin filaments are present, that is called as light band. This dark band is also called as A band. Light band is also called as I band. So in this myofibril, A band, I band, they are alternately present. And these dark filaments are made of myosin. Thick filaments are made of actin. The molecular weight of this actin is very low, whereas the molecular weight of this thick filament is more. So chemical structure is also different. And I said, in this dark band, in the middle region where actin filaments are absent, if you observe carefully, up to some extent, into the dark band actin filaments are protruded. That means up to here. This zone will appear very dark. That is due to the presence of both myosin and actin. The area of dark band where actin filaments are absent, that is called as H zone. So what is H zone? That is a central part of dark band. That H zone lacks actin filaments. On either side of that H zone, in the dark band, both actin and myosin filaments are present. So here, if you understand this, in this dark band, two areas. If actin and myosin filaments are present, both, that will be more darker than the central area. The H zone is without actin filaments. So, the general pattern of the arrangement of actin and myosin filaments are like this. Now, what we have to do? We have to understand the complete structure, ultrastructure of this actin and myosin filaments. Now, let us see in today's class what is the structure of actin filament. Whatever you are seeing here, that is not true. Just I am showing a thin narrow line to express that actin, thick line to express myosin. But under the electron microscope, that actin filaments will appear differently. Actin filaments. Now you have to listen carefully. This actin filaments are filamentous structure which is formed due to the accumulation of small units. The smallest unit of actin filament is called as G-actin. The G means globular. Globule means spherical. So I will show G-actin here. That is a small globule. This is G-actin. This is the monomer. Monomer in the sense single unit. That type of G-actins will gather, will attach in the presence of magnesium ions. So with the help of magnesium, this type of G-actin, one, second G-actin, third G-actin, like this, so many G-actins will attach with each other. I will show that also. This is one G-actin, this is one G-actin, this is another. So here in this diagram, I am showing 5 G-actins. Because of the attachment of 5 monomers. This is monomer. What is the meaning of monomer? Single unit. Like that, here 5 units are attached to form one cylindrical structure. So if you see from distance, this appears like a filament. See, that type of polymer of G-actins. What is the meaning of polymer? If that monomers attach with each other, it comes in chemistry. Polymer, monomer. Actually, protein is the polymer of amino acids, peptides. So like this here, if 1,000 or 2,000 G-actins attach with each other, at that time, one long filament will form. That polymer of G-actins are called as F-actins. What is the full form of this F? Filamentous. What is the full form of G? Globular. So this is globular structure. This is filamentous structure. So now you have to understand one thing. The actin filament is nothing but the more polymer of G-actins. In this actin filament, we can find many G-actins. If it is appearing like a chain, that is called as F-actin. Now, in one actin filament, in single actin filament, we can find 2 F-actins. 2 F-actins in the sense, they are coiled with each other, like this. That is just like the helix. They are twisted. Now here again I am showing in a form of single line. Actually, that is nothing but polymer of G-actins. Now you understood the structure of actin filament. So like this, we can observe, we can find 2 F-actins. This is one F-actin, this is one F-actin. With the combination of the 2 F-actins, one actin filament is formed. In this diagram, I am showing a single line, but internally, that single line is made of 2 F-actins. Each F-actin is made of many G-actins. You understood now? So, actin filament is made of two F actins. Each F actin is made of many G actins. So, one subfilament in this structure is F actin. In each F actin, one unit which is spherical is called as G actin. I think you got it. Now, this is not the only structure. In actin filament, along with these two F actins, we can find two more filaments. The two more filaments are present like this. Now, totally, you can observe 4 filaments in this. Out of that 4, 2 I didn't explain. 2 I explained. Which I explained? F actins. Now, what are these 2 new filaments in that? That filaments are called tropomyosin filaments. Tropomyosin filaments. So, totally now I will tell you, in one actin filament, 4 filaments are present. 2 are F actins, 2 are tropomyosin filaments. You understood? All the 4 are coiled with each other, twisted with each other. Now, what is the importance of these tropomyosin filaments? Tropomyosin filaments will help in attachment of actin filament with myosin filaments. You can see in the dark band, you can see in the sides where actin and myosin filaments are overlapping up to some distance. In that area, they must have some bond, they must have some relation. That attachment of that myosin and actin depends upon tropomyosin filaments. If the tropomyosin filaments are active, then this actin filament will attach with thick filament. I am not talking about thick there. That is completely thin filament. But here, that association is necessary. That attachment of actin and myosin filaments, that is necessary. So that's why for that binding ability, tropomyosin filaments will help. If we remove tropomyosin filaments, actin filament will never attach with myosin. No muscle contraction. So for muscle contraction, this actin and myosin must have some association link. That link is brought by this tropomyosin filaments. Okay, now you may get one doubt. How these tropomyosin filaments will attach with myosin thick filament? So, these tropomyosin filaments have some active areas, active sites. That active site I will show. This is one active site. This is one active site. This is one active site. So, in the regular intervals, in the series, throughout the length of tropomyosin filaments, we can find that active sites. I am showing 3 units in that structure. So, this is one active site, this is one active site, this is one active site. Such active sites are absent to F filaments, F actins. So they are present only to tropomyosin filaments. Now what are these active sites? What are the units? That units are called as troponin units, troponin structures. So now you are getting 3 names here. One is actin, tropomyosin, troponin. This troponin can be expressed as TP, troponin. We can write in the form of TP, troponin. In each troponin complex, there are 3 subunits. You can see the 3 spherical structures in each unit. That units are called as TPT, TPI, and TP. One is inhibitory, another one is troponin and tropomyosin. Myosin. So like this, 3 units are present in that area. So each structure, troponin structure, will have 3 subunits that they do different work. So now, if this tropomyosin filaments are present, if we remove that active sites in the sense troponin, this filament cannot attach with myosin. So in the other areas of tropomyosin filament, it cannot attach. So only in this spot, it can attach with myosin. So that's why you have to remember this structure. For understanding about the muscle contraction, you must have clarity in the structure of myosin filament as well as actin filament. So now let us see the structure of myosin. Myosin filament. This myosin filament is made of meromyosins. See, this is one monomer. This is the monomer of actin filament. This is the monomer of myosin filament. So each monomer of myosin filament is called as meromyosin. 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 That each monomer of myosin filament is having 3 areas. This region is called as tail. This broad spherical structures are called as head. And the middle region is called as cross arm. Cross arm. So this tail is connected with head through cross arm. So now, if many monomers join, it becomes polymer. In that attachment of 2 or 3 or 4 monomers, the tail part of many monomers will attach with each other to form one filament. That filament you are seeing here as myosin. So you are observing myosin filament in this diagram. That is nothing but the tail region. What about the cross arm and head? If you want to see that, you have to, here what I did, I shown one thick filament. This is very thick because there is myosin filament. Now this is formed here, this is shown here like this. But if you see, that filament is the attachment of many tail regions of many meromyosins. What happened to that cross arm and head? That will be there like this. That I am not showing here. in the mechanism of muscle contraction, we have to study about that. So at the time of muscle contraction, if this is the thick filament, up to this extent, actin filament is extended into the dark region. So when this head of each meromyosin becomes active or exposed, energy must be available. So at that time, this head will attach with this actin filament. As a result, one bridge is forming between actin and myosin. We call it as cross bridge. Before attachment with actin filament, we call it as cross arm. When the cross arm and head attaches with that actin filament, it appears like a bridge between actin and myosin. That's why cross bridges will form. So due to some movements of the cross bridge, actin filaments will be pulled towards M line. That I will explain later. Now, you have to understand about the structure of myosin. So, which is the monomer of myosin filaments? Meromyosin. How many regions are present to that meromyosin? 3 regions. One is head, another one is cross arm, and the terminal end of that each meromyosin is called as tail. The tail of many meromyosins will form the thick filament. On the surface of that thick filaments, we can find cross arms and heads of many monomers. So, to conclude, one thing I can say, actin filament, that is thin filament, is the polymer of 2 F-actins and 2 tropomyosin filaments. To the tropomyosin filaments, troponin complexes are present very frequently. Each troponin complex will be having 3 units. So they work differently, I will explain. So this actin filament is the combination of 4 chains. 2 are F-actins and 2 are tropomyosin chains. The troponin complexes are present to tropomyosin filaments. Now similarly, thick filament is nothing but the polymer of meromyosins. Meromyosin is like this. It is having twisted tail, cross arm. Cross arm is elevated and that is raised up, bearing head region. This head region has active site which can bind with actin. Here I shown that will bind with actin sometimes. So that's why we can call this area of that head of meromyosin as actin binding site. Actin binding site. We can call this troponin complex as myosin binding site. So when I say this is actin, this is myosin, if cross arms are present like this, here we have to show troponin complexes. Now you understood that head of meromyosin must attach with troponin complex of actin filament. Like that, if it is happening, why can't we call this as myosin binding site? This is called as myosin binding site. This is called as actin binding site. So you understood, if actin and myosin filaments are attaching with each other, in which areas they will contact. Now you understood that. Okay. So in this area, the myosin filaments will attach. These spots will attach with this area, head region of myosin filaments. So just copy that. And in some books it is given as this head as heavy meromyosin, tail as light meromyosin. So you can say this tail as LMM, light meromyosin. Head as HMM. What is heavy meromyosin. You copy the diagrams, I will explain. Right. So one addition in these three subunits of this troponin complex, one will help in attachment of calcium also. So in some books you can find it as TPC. One will help in attachment of calcium. Right now, just with this understanding, let us see how the muscle works, how the muscle contracts. If you have any doubts or not understanding, not collecting the information, don't worry. We can see again. I will explain. Right. So previous to this diagram, before explaining this diagram, I explained another one, that is muscle fiber, sarcoplasm, sarcoplasmic reticulum, highly branched, nuclei. I said if any cell is having many nuclei, that is syncytial condition. Syncytial cell. So muscle cell is example for syncytium in humans, multinucleated. Now along with this, myofibrils. When I explained about sarcoplasmic reticulum, I said one point there. That is the storehouse of calcium. So it will hold calcium ions. Now this is one muscle fiber. That muscle fiber, if it wants to contract, and one more reminding, this muscle fiber is related to which muscle? Skeletal muscle. Skeletal muscle is voluntary. What is the meaning of voluntary? If nervous system is controlling the functionality of that muscle, or if the muscle contracts and relaxes under the will of that individual, means if I am willing to show movements in my hand, then only my hand will move. If I don't want to extend, my hand will not move like that. So the movement of hand is under my control. So if I want to extend or if I want to bend, the information must come from my brain, my central nervous system. My brain has to send some message to that skeletal muscle present in my hand, then only it must contract to bend my elbow. Through what? Which route? That message will reach this hand from the brain? Nerve. So one nerve must innervate this myofibril, muscle fiber. If the nerve is coming in this direction, it is taking some message or nerve impulse to this muscle fiber. At the end of this nerve, we can find synaptic knobs. You know the structure of nerve, neuron. So dendrites will be there, cyton will be there, axon and terminal knobs. At the end of axon, we can find synaptic knobs, vesicles. So these are the structures. So here what we can observe in this area, there is a junction between the nerve and muscle. That's why we call the junction as NMJ, neuromuscular junction. So that is the place where the neuron or the nerve meets that muscle, neuromuscular junction. What is the message? What type of message is coming? That message is towards the contraction of this muscle. So that message reaches this synaptic knobs. At that time, synaptic knobs will release some chemicals on the surface of sarcolemma. The sarcolemma must have some receptors. What is the meaning of receptor? The structure which receives. So that receptors are present. Only that receptors will collect the information. Whatever the information came through the nerve, whatever the chemicals are secreted in that area, that chemicals can be identified by that receptors. So these are receptors. When the receptors are stimulated, activated by that chemicals, immediately this total membrane, which membrane? Sarcolemma, plasma membrane, will get depolarized. That means it will change the mode of permeability. Actually, you know, you studied in botany, all the plasma membranes of every cell is selectively permeable. That will allow some substances to pass in, and it will allow the substances, some particular substances to move out. So that means the plasma membrane is permeable for only some substances, not all. That's why selectively permeable. But when the receptors are activated, when the receptors are stimulated, at that time that will lose polarity. If it loses polarity, we can call biologically as depolarization, removal of polarization. What is polarization? What is polarity? The tendency of the movement of substances from one surface to another surface through any membrane. That is called polarity. So it is losing polarity. That means denature. In common words, we can say that nature of the plasma membrane is disturbed. When that is disturbed, the signals will, for example, in one family, if one person is disturbed mentally, automatically the other members in the family can recognize that. What happened, why you are very dull? They will identify, they will recognize, and they will ask. So like that, that depolarization will spread to the entire muscle fiber through sarcoplasm, because sarcoplasm is the matrix, that is the fluid. So that's why this depolarization will move to all areas. The complete length of this muscle fiber will understand that some message reached to the surface of sarcolemma. Now, in that sarcoplasm, sarcoplasmic reticulum is one structure that will also get depolarized. At that time, this sarcoplasmic reticulum will release calcium ions. So the calcium ions are released out by sarcoplasmic reticulum into sarcoplasm. So the calcium ions will be very free. When the calcium ions are released, immediately, what are these filaments? Myofibrils. What is that myofibril? This one. So definitely, the calcium ions will touch this each myofibril. All the myofibrils will become active. That means this structure will become active. When this structure is becoming active, within this myofibril, what is present? Actin and myosin filaments. Myofilaments. Actin and myosin. Just now I explained in the structure of actin and myosin filaments, there are some active sites or binding sites. In actin filament, we can see myosin binding site. In myosin filament, we can see actin binding site. They are very blind in resting condition. So when the muscle is relaxed, when the muscle is resting in state, at that time, that active binding sites of actin and myosin both will be covered. They are not exposed. Now, whenever the calcium ions are released from endoplasmic reticulum, that is sarcoplasmic reticulum, into sarcoplasm, at that time the mask will be removed. When the mask is removed for all the binding sites of both the filaments, they become very active. At that time, in this area, the peripheral regions of dark band, these active sites or binding sites of actin and myosin, both will be attached. So when they attach with each other, we can find physical contact between actin and myosin filaments. If this is actin, this is myosin. Myosin is thick filament. On the surface of actin, what is present? Troponin complex. On the surface of myosin, what we find? Cross arms and head, like this. I don't know whether you can see this. This is the head. This is the troponin complex. Now, when this calcium is released abundantly, at that time, the mask of both these two binding sites will be removed. They become very active and they bind with each other. For the binding, energy is required. So that energy will be collected by ATPs. That's why the head of myosin, meromyosin, which is called as HMM, that is also called as ATPase site, because it will be having some enzymes for that removal of terminal phosphate from ATP. So it becomes ADP and the energy can be released. So with that help of energy, it will attach with actin and that cross bridge will bend towards M line. Where is M line? At the middle of thick filaments, M line. So this cross bridge will bend towards M line from either side. So if actin filament attaches with myosin filament, if the cross bridge is bending, this actin filament will be sliding over myosin filament. If the actin filament slides over myosin, you can understand what is the meaning of sliding. Sliding means overlapping and moving one upon the other. So the same thing happens here. If the cross bridge of, if the connecting links between the actin and myosin bends towards the M line or towards the middle of dark band, the actin filaments will be pulled towards dark band. That means here previously the actin filament extended up to this area. Because of pulling force, the actin filament will reach up to here. Due to that, inward movement of actin filaments towards the dark band, H zone will reduce. What is the meaning of H zone? The area of dark band with only myosin filaments. Now the actin filaments are encroaching and entering towards the center, that means towards the M line. That's why that H zone reduces. If I say H zone is reducing, why that H zone is reducing? Because of inward movement of actin towards the M line. That means what happens to jet line? Jet line is also pulled because all the actin filaments will not stretch, that will move. If it is having elasticity, due to pulling force, it may become lengthy. That is not happening. That actin filaments are not having that elasticity. So because of the pulling force by myosin filaments, actin filaments will physically move inwards towards dark band. So the complete jet line will be pulled from either side. What happens to the length of this sarcomere? Yesterday I told you sarcomere is the unit of muscle contraction. That means the area between the two successive jet lines is called as sarcomere. So that complete sarcomere becomes short. If that sarcomeres become short in every case, because this sarcomere is repeated again and again, all the sarcomeres will become short. What will happen to the complete length of muscle fiber? It becomes very short. That is nothing but muscle contraction. Okay, just try to understand that. Tomorrow again I will revise. Thank you.